Miguel Soares is PI of the Information Lab at IGC in Portugal. And so today he's going to talk to us about his work. So thank you for accepting our invitation. Go for it, Miguel.
Thank you. Let me try to see. Whoop, that's not what I wanted to show. Can you see my screen?
Yeah.
Perfect. Okay. Well, thank you very much for inviting me to present our findings here. I was just realizing when I was putting the slides together that the findings are really a combination of the work of a lot of people. They certainly are, to a large extent, the work of Symnima Singh, but Symnima, who did the PhD with us, but she came after Bati Vilmat, who is also here, and I'm also still going to present some data from his own PhD. So it's a pleasure to do this. So I'm going to tell you a title that we've been following for quite some time about how mutations in glycans can really alter the rules of symbiotic interactions. So glycans are something that we don't usually study because they're kind of difficult and complicated. They actually core structural and exert modulatory functions. They're really critical. Sorry, I'm not managing here. Yeah, they're really critical for cognate recognition among cells from the same species or from different species. And the fact that cells from different species communicate very often via glycans makes them center stage for host microbial interactions. So this also sets into gear a series of coevolution processes. For example, if a host evolves to alter a glycan or the structure of a glycan, microbes co-evolve to recognize that altered glycan. They can also evolve to avoid decoy glycans that are sometimes generated by hosts, or even, as you will see, mimic the glycans that sometimes are expressed by hosts. So what I'm going to tell you about is a host that evolves to delete glycans and microbes that evolve to mimic those glycans. And one thing that happens that it's not often taken care of is that if a mammalian loses a glycan, that glycan stops being a self-glycan, and now you can get immunity against the glycan. And you will see that this will have implications. So the gene that we're going to talk about is GGTA1, is alpha-galactosyl transferase. It produces two GALs linked via 1,3 link, which is here. And we usually represent them like this. And the hosts that we're going to talk about are humans, ultimately. But the way to understand this is that New World monkeys, including, for example, lemurs, they have a functional GGTA1 gene. But Old World monkeys, including humans, have this mutation that appear very early on, and two other mutations that added to it and makes that we have a non-functional GGTA1 gene. And because of that, we don't express the end product, which is alpha-gal. So how did this loss of function, as it occurred in primate evolution, modulates host immunity? So we're going to be more specific now. We're going to talk about humans that carry loss of function mutations in this gene, and specific bacteria that now carry homologs, orthologs of this gene, and can express actually the glycan alpha-gal. Now, but humans have anti-alpha-gal specific antibodies, and so this will have implications. So just to remind you very briefly, these are the New World primates, the lemurs. They have the development of B cells. It doesn't quite matter exactly how this works, but these are B1s and B2s. They're the two flavors of cells, if you will, that occur. They develop normally to generate peripheral cells that can produce IgM. In New World primates, sorry, So New World primates express alpha-gal, and therefore they delete the peripheral B cells that express alpha-gal, because this is a self-antigen, and they also delete these B2 cells. But we, as members of the world primates, do not express alpha-gal, and therefore we can generate B cells all the way until the production of alpha-gal reactive B cells. Now, do we have components of the microbiota that express alpha-gal? And the recent manuscript suggested through genomics that indeed this is the case. We screen a fairly large panel of components of the gut microbiota that are used as probiotics by a company. And we find that some of them express fairly high levels. You should keep attention to this one because this one is going to be important. And this is just a series of flow cytometry assays in which you see in green that indeed these bacteria are expressing alpha-gal compared to the controls, and they express different levels. And it appears to be fairly homogeneous. So they can express orthologs of GGTA1 that express this alpha-gal glycan. So we start wondering what would be the effect of this. And with Bati Vilmas, what we did is to actually colonize mice that are in a way humanized because we make them lose the GGTA1 gene, and ask what would happen if we now colonize them with a bacteria that expresses alpha-gal or not. And what we see is that when we colonize them with a bacteria that expresses alpha-gal, they now produce IgM against alpha-gal. Now, this is very similar to what happens in humans, and in particular, we focus on this population. You're going to understand why this is, which we followed over time and over age. And you see that we are born with very little anti-alpha-gal antibodies, and then we increase over time. And this population in Africa here has much more than, for example, Americans. So the people in the United States and the East Coast. So that means that environmental factors, presumably including food that influences microbiota composition, will play a very strong role on the production of the steady state anti-alpha-gal antibodies. So what is the impact of these antibodies on the microbiota? So what we did here is to take mice that are again humanized because they lack alpha-gal, or they are normal mice that express alpha-gal, and we look at the microbiota. And what we found initially, to our astonishment, that they were very, very different for unweighted unifrac and for weighted unifrac as well. And we followed this up and did a LabC analysis and actually identified or start identifying some of the classes of these bacteria that are favored, for example, in GGTA1 deficient mice. And we see very often proteobacteria appearing here. So what we did here is to cross the two genotypes such that they inherit the microbiota from the females. So we get heterozygotes in F1, and then we segregate the genotypes again, but now they all carry the female microbiota that came from a GGT1++. So now here we have the two genotypes carrying the same microbiota. We checked for that. So this was the F0 here. This is the F1, and this is actually the F2. These guys here now, they have a normalized, let's call it microbiota. So when we do this and we keep actually maintaining the genotypes separate between themselves through several generations until here, until F4, we see actually that the microbiota starts segregating. So it doesn't mean that it's really the genotype that is imposing the segregation of the microbiota composition, but it's suggestive of that. Now, what we did afterwards is to ask what would be the impact of this microbiota on the immune response. So such that if in the absence of alpha-gal, the GGT1 knockout mice are shaping their microbiota, we thought, or we hypothesized, that this might occur via the production of IgA antibodies that can actually transmigrate across the epithelium barrier. And in these experiments, what we saw, actually, they were performed by Jess Thompson. We see actually that a mouse that expresses alpha-gal has about 8.6% of the microbiota in the small intestine that is actually IgA positive and expresses alpha-gal. So these are immunogenic bacteria that express alpha-gal. And we start noticing actually that in the GGT1 knockouts, this population tended to decrease. So Jess quantified that. And indeed, she found that there's a very significant decrease on the percentage of alpha-gal expressing bacteria, on the percentage of immunogenic bacteria that express alpha-gal, but not in the overall percentage of immunogenic bacteria. So it looks as if once you lose alpha-gal, you're now able to target this alpha-gal expressing bacteria by AGA and presumably change the composition of the bacteria. We also asked whether having or not having this gene would have an influence in the way you react to a microbiota, in this case, that comes from an alpha-gal knockout mouse. And we found that after colonization of germ-free mice, there were either GGT1 knockouts or wild types. The knockouts actually did a stronger IgA response, suggesting that they have somehow a better ability to produce IgA against the microbiota, which would justify why they are shaping the microbiota. We don't see differences for IgM production, and we don't see differences for IgG production. So it seems to be rather specific to alpha-gal expressing bacteria. Now, to really prove that it's B cells and antibodies that are shaping the microbiota of the GGT1 knockout mice, we did a really complicated experiment in which we now have mice that are GGT1 plus or minus, and when you see the red here, we're knocking out the B cells, so they cannot produce antibodies. So we segregated all the genotypes and maintained initially we controlled for the microbiota, and then we segregated the genotypes, and we asked whether the microbiota would be segregating in a way that would be dependent on B cells and or the GGT1 genotype. So what we see in unweighted unifrac, these are a bit difficult to grasp in the beginning, is that in the presence of B cells, there is actually a difference between this genotype, but it also appears in the absence of B cells. Now, interestingly, if we go on weighted unifrac, this difference is only significant if you have B cells. That means that losing alpha-gal shapes the microbiota via a process that requires B cells, and it disappears actually if you don't have B cells, even if you're changing this genotype. So this to us is part of the proof that actually when you lose alpha-gal, you're shaping the microbiota via a mechanism that is dependent on B cells. The distance between in the PCA analysis between the plots for unifrac actually are more distant if you are knockout. That means that having or not having B cells imposes a bigger difference if you are in a knockout background, and here seems to be the reverse, but these are the low abundance. So for the high abundance and the structure of microbiota, it seems again that having or not having alpha-gal depends on having or not having B cells to provoke these differences. Now, what we see again is that we see the appearance of bactericidae, and we see actually that they decrease once you have antibodies in an alpha-gal background. So it looks like part of this antibody response is containing the expansion of this specific bacterial class. Now, this is just another way to show you that, and it shows you that actually they're being reduced here by the presence of B cells in a GGT1 knockout background. So what I told you in a fairly quick way is that once you have alpha-gal specific antibodies, you can now interact with your microbiota and eventually shape it. Now, are there other things that occur by knocking out GGT1 that would be of relevance? So one of the things that Bati Vilmas found was that this carries a big impact on malaria. So very quickly, malaria is a vector-transmitted disease. It's transmitted by Anopheles mosquito. It drops sporozoites in the skin. They go into the liver, then they go into blood, and eventually they will be transmitted. So we got very interested at this stage here. And the reason why we got interested on this, it's because Bati found that Plasmodium, both Falciparum, Bergae, or even Ueli, express actually a mimicry of this glycan. So they express the alpha-gal glycan. So the same glycan that is in bacteria is now being expressed in sporozoites. Now we went back to this population that lives in an endemic area of malaria, And our colleagues at NIH found that actually individuals that do not get infected through a wet season in an area that is endemic for malaria, they tend actually to have higher levels of anti-alpha-gal IgM antibodies than those that get infected. This is a very minority of the population because almost everybody does get infected. So Bati tested this functionally. So we colonize the mice using the same approach that I told before with bacteria that express alpha-gal or not. We wait for the antibodies, for the colonization to occur and for the antibodies to rise. And then we are actually exposing them to Plasmodium sporozoites that express the glycan, and we ask for Plasmodium transmission. And what Bati found, I would say to our astonishment, was that the E. coli 086 B7 that expresses alpha-gal is now protecting against transmission. One would not expect a full protection because humans still get protected, but it's a very significant one. Now, if you take out IgM from these mice, remember I told you that once you colonize with this bacteria, you now have IgM against alpha-gal. So with this mutation, it allows to take out the IgM. And what Bati found is that the protection disappears. So this allowed to identify there is IgM antibodies against alpha-gal that are protecting against malaria. So we can add another arrow to this to say that another complexity that appears here is that you're shaping your microbiota, but you're now all protected, also protected against third-party pathogens that express the glycan. So it's going to get a little bit more complex, but I think interesting. Are there any associated advantages? Since we all lost the expression of this gene, this must give us real strong advantages. So here we normalized against the microbiota, the microbiota of these two genotypes, bringing the microbiota from a female GGTA1 knockout. We have F0, this is the F0, then we have the F1. And then we compare it in F2 outer war. We verified that indeed they now normalized, both for weighted and unweighted unifrac. And then we subject them to sickle ligation and puncture, which is a very well-established approach to induce sepsis. So we puncture the cecum, and the bacteria, they are now normalized between the two genotypes. They gain access to the peritoneal cavity and they cause sepsis. And what SymbNET found was, actually this work was even initiated by Bati still, but SymbNET confirmed, and this experiment was actually done by her, is that under the same microbiota, there's a survival advantage of not having GGTA1 allele. So this was interesting to us, and SymbNET associated this to a very strong reduction in microbial burden, so in pathogen burden in peritoneal cavity, blood, spleen, and lungs. So it's at least 1 log. So it seems to be a resistance mechanism by which these animals are protected. So we went after this and started asking, we thought it was going to be very simple. Why do you become protected? So here what we took is a mouse that lacks adaptive immune system, no B cells and T cells. It also lacks GGTA1. So it's like, a bit like a human. And we took actually antibodies that we purified from the circulation of mice that we found to be protected against sepsis or not protected against sepsis. And we took IgG and we transferred to these ones. And then we infected them with the cecal slurry, which is just an inoculum from the cecum, but to maintain exactly the same. And we saw whether the antibodies coming from one genotype or the other would exert different effects. And the effects were very dramatic. So the antibodies that were coming from the protected animals against sepsis are very protective, the IgGs, and the other ones are not protective compared to a control. So there's something special about these antibodies. So we took the antibodies and we went on and analyzed for different aspects: IgG subclasses, ability to recognize bacteria, or ability to recognize alpha-gal. For the subclasses, you can see here from the different sources. So this is the ones that protect, this was the ones that don't protect. They, in both cases, gamma 2b. So this is a very good complement Fc gamma receptor binding antibody isotype. Now, for their ability actually to recognize alpha-gal, they actually recognize very little alpha-gal. And this, I'm sorry, this is the amount, the total amount of gamma 2b that there is in the inoculum, And this is the amount of that gamma-2b that can recognize alpha-gal. So very residual. If we compare them for the ability to recognize alpha-gal expressing bacteria in the microbiota, it's again very, very residual. And it's just quantified here. And we then start comparing them broadly for their ability to recognize different components of the microbiota. And they can recognize, but to the same extent. And if you focus here, for example, on E. faecalis, it turns out that E. faecalis does not even express alpha-gal. So the ability of these antibodies from a GGTA1 knockout to protect against bacteria, it's somehow independent of their ability to recognize alpha-gal. And they seem to recognize bacteria in a similar manner to the antibodies that are not protected. And what really convinced me to do this is actually an experiment that was proposed by Gabriele Victoria. So here what we have is the same bacteria inoculum, and we're putting AGG that comes from the knockout. This is the AGG that protects and the AGG that does not protect, but they recognize the same thing with the same proportion by flow cytometry. So they don't seem to have any difference in their ability to recognize bacteria, yet one protects and another one doesn't protect. We pull down this bacteria to see if we would find any changes, and weighted unit frac the same, weighted unit frac the same. We asked for Bati Vilmas already. He had been already in Andrew McPherson's lab for quite some time. They had developed actually a way to analyze for the AGG repertoire of mice. So we asked them whether they would compare the two genotypes, and actually they find exactly the same thing. They cannot segregate these mice according to their IgG repertoire in any way, shape, and form. So same repertoire, same ability to recognize, but very different in the protective effect of these IgGs. So very puzzling, and we turned into an alternative hypothesis, which was to say, well, perhaps the problem or the issue with the alpha-gal is the alpha-gal that is expressed on the IgG itself. So we became aware of this band, not aware, but we became very interested on the structure of these B-antenary glycan structures that fall into IgGs, including on gamma-2b. The hypothesis is that a wild type should have alpha-gal, but a knockout should not have alpha-gal here. So Nima actually purified the IgGs and looked for it coming from the two genotypes. And indeed, we see alpha-gal being expressed. I'm sorry, this is the, yeah, sorry, I'm sorry for this. So this is an anti-alpha-gal antibody that we used, and we can see both avian light chain being actually expressing alpha-gal from the wild type, but not from the knockout. And this is just to show you that the protein loading is the same. So once you express the GGTA1, indeed you express alpha-gal, and when you don't express, indeed you lose the expression. So we thought that this might have an effect on the effector function of the IgGs. So we tested for that. Jessica Thompson did that for the activity of neutrophils. It's a tough technique. So we put them in the peritoneal cavity. We pre-incubated with the antibodies from one or the other genotype. And from the GGTA1 knockout genotype, they appear to have a slightly increased effector activity against bacteria, which would be consistent with a slightly increase in the effector function. Now, we thought that this might be related to Fc gamma receptor interactions. We know that different subclasses can interact with different Fc gamma receptors. We focus on this one, and we tested them all. And what Sunima found is that indeed, the IgGs from the knockout, they have a higher binding affinity, as you will see in a second, against the Fc receptor compared to the IgGs from the wild type. They don't show any difference in a series of controls, and they also don't have any difference in their ability to activate complement. So actually you measure this affinity, and you see actually that there is an increase from year to year from 54 to about 40. So we're really sure that the interaction of the IgGs that come from alpha-gal knockout have a slight increase on Fc gamma receptor affinity. So what this says is that in addition from shaping your microbiota, becoming protective against pathogens that express alpha-gal, when you lose GGTA1, you also increase IgG effector function, and that can give you protection against bacteria, irrespectively of expressing alpha-gal. So one way of finishing this is to say, if this is so good, why don't all mammalian species have a deficient GGTA1? So there must be a fitness cost associated with this. And through a heroic enterprise, SymbNET actually analyzed breeding pairs between GGTA1 wild types and knockouts over 4 to 5 years to try to see whether there was a difference. And what she found is that initially, during the first 20 ages, we don't see any difference, but over time, the knockouts actually stop breeding and produce less litters compared to the wild types. And if we look at this in a cumulative way, it's actually very significant. We go from about the time of reproductive senescence is reduced by about almost 50%. So not having GGTA1 somehow precipitates reproductive senescence, and mice are not able to have as much as an offspring if they are knocked out for the enzyme. So that's the price that one pays, which is put here. Now, if the price is so big, would there be an advantage to lose this? So we collaborated with Emergence to do this, and she did a mathematical modeling of this trade-off versus the protection that you have. And the conditions of known infection, where the fact that you don't have alpha-gal actually reduces fitness by about 53%, which is what I showed you. But if you now impose and model conditions of high virulence and high prevalence of a pathogen, which is around here, so very, very high, now you can get a fitness advantage that overcomes the reproductive disadvantage. So in conclusion, the deletion of GGTA1 allows for alpha-gal specific immunity that modulates the bacterial composition of the gut microbiota. Immunogenic bacteria in the gut microbiota actually are targeted by alpha-gal immunity, and that reaction confers protection against third-party pathogens that express alpha-gal like glycans. Deleting GGTA1 from the IgG really changes the effector function in a way that you can actually change protection against pathogens irrespectively of the ability to produce alpha-gal. There's a trade-off to this, which is very high, which is a reduction in reproductive output, but mathematical modeling suggests that actually these things wear off, and you're better off even with this trade-off to lose GGTA1. So I'd like to thank all the sources of financing, but in particular, SymbNET and Bati that did the bulk of the work. Jessica Thompson, that I mentioned a couple of times, was also instrumental. So was Patricia. Sebastian Weiss also contributed in area that did the mathematical modeling. And I would like also to thank different core facilities at the IGC, without whom this would not be possible. I'll be very happy to answer any questions that you might have. Thank you.
Thank you so much for your presentation, Miguel Soares. Really nice talk. Thank you so much.
Thank you.
So again, we are open for questions. If the audience wanna ask, just raise your hand in the button. So I will start with a question, if people don't mind. I'm thinking about if the primates like ourselves are experience this loss of fitness, like if you can say that we're still feeling like experience this loss of reproductive fitness. Do you think we as like primate lineage have already overcome this situation or not?
I'm not entirely sure I understand your question, but I will try to answer, and you tell me if I'm.
Yeah, I can reformulate it.
I think So you asking whether we overcome that fitness cost. If that fitness cost exists in humans, we did, because we all ear, so it worked okay for us. But let me tell you another thing. So in the literature, it looks as if this reproductive senescence is a feature of old world primates. So it doesn't prove anything, but there's even the possibility that it's this mutation that imposed that fitness. This is us. I mean, there's literature to sustain this, but I'm not saying that this is the truth. So it would look as if indeed this was a fitness cost imposed to non-human primates. And we managed to overcome this because we survived and we have actually too big of a fitness. Now, what really drove this in the ancestral primates that gave rise to all the world primates, that's really a mystery. So the work from Bativilmas suggests that a pathogen like Plasmodium could do this. But it might have been something much nastier than Plasmodium, perhaps envelope viruses, zoonotic envelope virus expressing alpha-gal, extremely virulent and extremely prevalent. Because the incredible thing is that we carry 3 independent mutations that were selected. So there were at least 3 founding events. So this must have been, if it was a pathogen, a very, very nasty pathogen, that compensated according to the mathematical model for the huge fitness cost that we see imposed in mice, but we cannot do it in humans, of course. But we're speculating that might be the same. So, and there's other events that I would love to study that are not here, that give further advantage in selection and speciation.
Yeah. I see that you get a point. Another question related to that is if there is another example in mammals that you can find this kind of loss of function, because I'm thinking about that this kind of pressure could help happens in other scenarios.
Yeah, so that's a great question. There is another. There's a sialic acid that got mutated. Actually, it's one of the only distinctions between chimps and humans. It's the loss of NO5GC, which is encoded by CMAH. And it's much more difficult to find out what is the selective advantage of not having that. But it has to do, again, with altered immune responses. Has to do is a overstatement, but it has been linked functionally in mice by the group of Ajit Varki that works extensively on this with altered immune responses against some pathogens. And there's a trade-off there that appears to be cancer and tumorigenesis, but very, very stringent as well.
Great. Wow, it's an impressive work, a very interesting one. I took a look here in the audience and they don't have any question for now. I took a look at the chat and neither there. So I think I want to say thank you for you both, Miguel and Michal. And also thank you for the audience to be here with us in another SymbNET seminar.